My name is Michelle McCully, and I'm an assistant professor in the biology department. In my lab, we study the difference between naturally occurring and engineered proteins and the balance between their stability, their dynamics, and their function. Engineered proteins have the potential to solve many problems. They can serve as diagnostics, as vaccines, they can be found in household cleaners, or in industrial procedures. The potential applications for engineered proteins could be broadened if we were able to design proteins that worked at high temperature. For example, a diagnostic could work in a warm climate, or vaccines wouldn't need cold storage conditions. In our lab, we use a model set of proteins where one is functional but not thermostable, and the other is engineered to be thermostable, but it's not functional. We're hoping that we can learn general qualities about the engineered thermostable protein that will allow us to generally design proteins to be thermostable and functional. We perform simulations of these proteins to see whether or not they can perform their function of binding DNA or whether they can maintain their folded structure under high temperature. What we have found so far is that the functional protein is indeed able to bind DNA over the course of our simulations, but the thermostable one is not. We've also found that the functional protein unfolds at high temperatures, making it non-functional at those high temperatures, whereas our thermostable protein maintains its folded structure even at high temperature. We've found that the thermostable protein is unusually dynamic, and we think that in fact these heightened dynamics are what allows it to be thermostable. This is not a common property to see in proteins, and we think that we might be able to apply it more generally when we're trying to design proteins to be thermostable. So we're testing it in our system, and we're trying to pick out the structural aspects of the protein that make it thermostable, and then we're also trying to pull out the structural aspects of the functional protein that make it functional, so that we can build chimeric proteins that are both functional and thermostable. We perform simulations of these proteins to see if they can bind DNA and if they stay folded at high temperatures. And then for the candidates that look the most promising, we actually make the proteins in the lab and then we test them experimentally. When I say we, I mean a team of 17 undergraduate students who have worked in my lab over the past 5 years. These undergrads perform all parts of the scientific process. They design our experimental questions, they set up and perform and analyze our simulations, then they express the proteins in the lab and they run the assays to test for their function and their thermostability. They've presented their data at several national conferences and also in a few peer-reviewed publications.